Hi everyone, my name is Emma Sellers, and I chose to explore how artificial dyes affect the body and the brain at different levels of psychological and biological analysis. And my name is Emma Sellers. Artificial food dyes are human-made colorants created by chemical compounds. They can be found in candy, cereals, drinks, and even pantry house common items like spaghetti sauce. Although they are considered safe at regulated levels, research suggests that some dyes may influence behavior, attention, and neural activity. Psychologists care about food dyes because several studies link dye consumption to attention problems, hyperactivity, and cognitive changes. These effects are especially relevant to people with ADHD or autism who might be more sensitive to environmental factors that influence attention networks. Understanding the biology behind these behavioral changes helps explain why dyes might influence cognition. Biologists study food dyes because they can affect the body at multiple biological levels. Some dyes cause cellular stress, some others trigger inflammation, and some influence neural activity. These biological changes can affect how the brain regulates attention and behavior. One of the studies that I use, by Kirkland et al. In 2022, examined how artificial food coloring affects college students with ADHD. They used EEG, which measures electrical activities in the brain, to study attention using theta and beta waves, which reflect cognitive control. After consuming artificial food dyes, participants showed increased theta power and changes in the theta beta ratio, a known biomarker for attention difficulties. Participants also reported worsened ADHD symptoms. This gives us a system level view of how dyes can alter neural activity related to attention. Okay, for my levels analysis, I will be doing the cellular level, the networks and system levels, and then the whole body system level. At the cellular level, Muranos, Amo, and others found that several common food dyes cause oxidative stress, DNA fragmentation, and reduced viability in both in vitro human cells and in vitro fruit fly models. These findings matter because neurons are especially vulnerable to oxidative stress. When neurons experience cellular damage, they their ability to fire efficiently is reduced. This can interfere with the precise timing needed for attention and cognitive control. At the whole body system, Anchava and others described how dyes are absorbed and metabolized. Some dyes can trigger inflammation, immune activation, and metabolic stress. These responses matter because inflammation can influence the brain through the gut-brain axis. When the immune system is activated, inflammatory molecules can differ how effectively neurons function. This provides a pathway from dietary exposure to changes in brain activity. Finally, at the networks or system level, the EEG findings from Kirkland et al. Show how these biological changes translate into altered neural activities. Increased theta power and changes in the theta beta ratio suggest that attention networks, especially those involving the prefrontal cortex, are functioning less efficiently. When neurons are stressed or when inflammation affects signaling, attention networks become noisier and less coordinated. This matches the behavioral reports of worsened attention after dye consumption. When we combined all of these findings, we see a clear pathway. At the cellular level, dyes cause oxidative stress and DNA damage. At the whole body system level, dyes trigger inflammation that influence the brain. At the networks and system level, neural activity becomes less efficient, affecting attention. Together, these levels explain how food dyes influence attention and performance. Thank you for listening, and I hope you enjoyed learning about artificial food dyes as much as I did.